All right. Thank you, Jeremy, and the organizers of the meeting for the invitation to speak. So I'm going to tell you a bit of a different story now, and we're going to do a bit of a deep dive, a little bit into biochemistry and back out and see how that connects with omics. And so the title of the talk, Understanding Plant Cell Wall Biosynthesis to Improve Plants for Biofuel Production, but the last part, the power of a combined fundamental and directed approach. It's a point I want to make at the beginning. It's absolutely clear from the JGI showcase, from the talks we've heard and the ones we will hear, how powerful various omics techniques are, genomics, transcriptomics, metabolomics, and others. But I'm a firm believer that you can derive the most power from those when you combine specific questions with an expertise in certain domains because of the complication of the gene products. And maybe that will come through in this talk. All right, each year, 100 billion tons of carbon dioxide are fixed by plants, marine and land plants, into carbohydrates. Now, we heard this morning an example of the human ability to form an enzyme that can fix carbon dioxide 20 times higher, but still plants do a pretty amazing job, and it's the basis of life, most life as we know it on this planet. The bulk of that carbon in that plant biomass resides in the plant cell walls. And the cell wall, obviously, is that complex carbohydrate-rich matrix that surrounds the plant cell. All right. Plant cells not only have many uses for humans, but they're multifunctional in the plants. And in the simplest sense, you can divide those cell walls into two parts. The first walls lay down, the primary walls, are interesting. It's this complex matrix that has to have the ability not only to give strength to the plants, but also to allow, when there's enough turgor pressure, for those cells to grow. So you've got to have mechanisms within that complicated matrix to allow slippage between those polymers. And that's something that many scientists are still trying to understand, and the controls of growth are still somewhat elusive to us. After that primary wall is laid down, in many cells that differentiate, that wall's pushed out, and there's a secondary wall produced that is different somewhat in its polymer structure, has more cellulose generally, hemicelluloses, and lignin. If you look now and take a deeper dive at what that cell wall is, and it is a carbohydrate-rich, carbon-rich extracellular matrix. It also, in secondary walls, can have lignin, and there can also be proteins, but the bulk of it is carbohydrates. What you see, whether you're looking at an electron micrograph of a primary wall from dicots like Arabidopsis or grasses, in this case corn, in all cases they're polymers. And if you look carefully at this, you'll see that those polymers in part seem to be interacting, and we do not understand well at all the covalent and non-covalent interactions that occur in those polymers and those that control growth. All right. As humans, we like to have models so that we can begin to try to understand and also have hypotheses we can test. And many of you who looked at cell wall models have seen models like this. There are three main polysaccharides that are constantly described in the wall. Cellulose, the reinforcing fibers, hemicelluloses, on the left, those are in red that interact or are shown to interact via hydrogen bonds with the cellulose. And then shown in all sorts of different ways and all sorts of different models are the pectins, which are blue ribbons here. If you look at a model that has more detail, again, you have cellulose, hemicellulose, and now you begin to get a little more definition in the pectins. You see that there are these kind of long polymer-like regions, and there are regions that are branched. And we're going to go into a bit of detail and then come back out to one of the main points I want to make in this talk. One point that I want to leave you with is the following. A simple 3 or 4 polymer view of the wall, I believe, is undercutting our ability to connect omics to function. And you'll see why as I go through this talk, I say that. All right. I have worked over 25 years, close to 30 years now, on studying plant cell wall biosynthesis, emphasizing pectins, but looking at some of the other polymers, really concentrating on biosynthesis and connecting that with structure and function to the wall. About 13 years ago now, I was fortunate to be part of the Bioenergy Science Center, BESC for short. And one of the goals, the main goal of BESC, was to understand, step back a minute, if we want to use the carbohydrates in the wall as an energy source for biofuels, you have to get them out. They're much harder to break apart because of that complex matrices. You have to use chemicals and high temperature to get them out. And we call that difficulty to extract the polymers to make them accessible to enzymes, recalcitrance, the recalcitrance of biomass. So the goal of BESC was to try to reduce recalcitrance. You could do that by improving the microbes, But what I was involved with, and about a third of BESC, was to try to improve the plants, to make it easier to break those polymers apart. And what we found is by connecting our expertise in cell wall synthesis to that goal, we've made some interesting discoveries that I'll outline some of those in a minute. Now, more recently, I've had the good fortune to be part of the Center for Bioenergy Innovation, and we moved on, and we made quite a bit of progress in BESC, and we had identified hundreds of genes and thousands of lines that had reduced recalcitrance, and identified a number of those that even had improved growth. I'm going to tell you about one or two of those. But more recently, in the Center for Bioenergy Innovation, we're moving behind that and taking advantage of natural variations and omics technology to try to more quickly take advantage of the genes in the population that can make a convergent improvement of the biomass. And I'll tell you a little bit about, connect the study to that at the end. Okay, so let's go back. So in our very early studies in the BESC, in the Bioenergy Science Center, as we were looking at transcriptomic data and trying to identify any pectin genes that might be interesting recalcitrance candidates, we identified several, pectin biosynthesis genes in switchgrass and populus. And that surprised us a bit because those bioenergy feedstocks aren't abundant in pectin. And I'll go through that in a little bit later. If we look at a table like this, where you're looking at the polymer, cellulose, lignin, pectin, hemicellulose, and the percentage that exists in various types of biomass, if you look at dicot primary walls, something like Arabidopsis leaf, poplar leaves dividing cells, they're rich in pectin. And it wouldn't be surprising if pectin plays a role there. It makes up around a third of the non-cellulosic carbohydrate. But in our bioenergy feedstocks, which are largely grasses, as has been discussed earlier, pectin makes up only 2%, sometimes up to 5 or 10%, but usually around 2% or less of the wall. And then in the other types of bioenergy feedstocks that are rich in secondary wall, pectin makes up less than 10%. So if you're trying to modify biomass, most people concentrated on lignins, hemicelluloses, and perhaps cellulose, but we began to identify the potential role of pectin in recalcitrance. So before I can tell you about those results, though, let's review, or maybe learn for the first time, what pectin is. Pectin is a class of polysaccharides that's defined as having galactronic acid that's hooked at the 1 and 4 position, okay? If you do a little bit deeper dive in the pectic polysaccharides, and this is the only slide I show this kind of structure on, the most abundant one is homogalacturnan. It's galactronic acid hooked in an alpha-1,4 linkage in a linear polymer. So it's just like starch, except instead of glucose, it's made of galactronic acid. That gives it some interesting properties. We're coming back to it, and I call this HG for short. The second most abundant polysaccharide is rhamnogalacturnan 1. It's like the glycosaminoglycans in your body, an alternating Galley-Rhamnose-Galley backbone, but it's very highly side-branched, and we'll mention it a little bit later. And then finally, the least abundant but most complex pectic polysaccharide is rhamnogalacturnan 2. It has a homogalacturnan backbone and 4 very complicated plus 2 additional, not shown here, side branches. This structure is almost conserved across all plants, and it's very important. Now, one other point I want to make about the pectins before I go on is that the pectic polysaccharides are cross-linked in the wall in at least 2 manners. One of these is that homogalacturnans can have ionic interactions between each other. So here we have one chain of homogalacturnan. Here's another chain of homogalacturnan. Now these are galactronic acid. I didn't tell you they can be methylesterified and that can be modified. What we're going to talk about enzymes that make the backbone. So when they're not modified, they have carboxylic acid, a lot of it, and so they'll interact with divalent cations and form these salt bridges, and that can be really important in cell adhesion. The other way the pectins cross-link is via RG2. So you have up top here one RG2 molecule with this side chain. Here's another one, and they make so-called borate diesters. This is really important. If you don't have those, you'll get dwarf plants, if you get plants at all. All right, we'll come back to this later. All right, now is an important concept. If you looked at those models, it looks like pectins, hemicellulose, cellulose, kind of boring. Yeah, they're made of sugars. Let's go on. So imagine you're doing omics now, and you identify a gene, and you say, oh, that's involved in pectin synthesis. And in your mind, this is simple. pectin polymer, okay, good, it's involved in pectin synthesis. It's not that simple. And what we've found through many years of research is the following. If you look over 60 years of structural research over how the pectin polysaccharides are connected, this is what you see. In red here, I'm showing you HG. HG can exist as a long polymer in the wall, but it can also exist as domains where it's cross-linked with regions of RG1. And don't forget, these RG1s are highly branched. And it's also connected to RG2. And then more recently, we've shown it can be a glycan domain in complex proteoglycans in the cell wall. So now what you should be thinking in your mind now is, oh, that cell wall is much more complex than those simple models suggest. And now if I am studying genes or changing genes, and I'm affecting something that complex, and I have multiple members in a biosynthetic gene family, I might be affecting different polymers differently. All right. So increasing evidence suggests, as I just said, that it's more accurate to think of pectin as domains in different wall polymers, and we only understand some of those wall polymers at present because it's difficult to get in there. So in my lab, the two questions that we concentrate from a basic side is how are the pectins synthesized, and particularly we started with HG, and how do you distinguish the genes that encode the different polymers with the different domains. Now you'll see why I'm telling you all this, and we're going to move rather quickly now into what this has to do with bioenergy and biofuel production. But in regards to using what we know on the basic side and applying it, the final question is, what role do these pectin domains have in growth and biomass recalcitrance? So in order to describe some of our results, I need to now finally introduce a gene family of pectin biosynthetic genes that we identified. And that gene family is known as the Gout gene family, short for galactoronosyltransferase, and the first gene that we identified was Gout1. So there are colored pictures and symbols you can use for sugars. So this represents homogalacturinan. I mentioned that alpha-1,4-link GAL-A polymer can also be methylesterified and acetylated, and that complicates its function, but that's the second stage in synthesis, and we're really interested in the first laying down of that backbone. So we set out to identify homogalacturinan galactoronosyltransferases, and we're successful, and identified the first one I'll tell you about in a second. It was a member of a 15-member gene family in Arabidopsis. Now in this phylogeny, I don't know how well you can see it there, there's a single black gene in each of these. These are 7 subclades, and the 15 genes are divided among them. That's the Arabidopsis gene. In red is the poplar homologue. There was a gene duplication, so most of the gouts are duplicated in poplar. And then you're seeing here rice, and later I'll show you the switchgrass, in which there was an expansion in many of these gouts, which should be a little surprising since pectin's not very abundant in grasses. So we identified some time ago Gout1 that synthesizes homogalacturinan, found out shortly thereafter it functions in a protein complex with Gout7. And that's fine. But if you really think back on what I told you before, where those H gene domains are, then you have to ask yourself a question. In which Gout1 synthesizes which HG containing polymers? Does it synthesize the long polymer? Does it synthesize HG connected to RG1? Does it synthesize HG connected to RG2? Or does it synthesize HG in a proteoglycan or some other HG containing polymer that we don't know? Now, to answer that question, you need to look into the plant. And so, I'll get to that in a second. So we've done studies on Gout1 and the Gout1, Gout7 complex to begin to answer the question of what polymer HG synthesizes. And Robert Amos, who's just finishing up his degree now, did a very nice study where he was able to express this complex in an intact form in human embryonic cells. And he did a lot of really detailed enzymology, but the only point I want to make here, he saw that the enzyme can produce 2 types of products. It can either with acceptors or by itself synthesize very, very long HG polymer, but it can also take an HG acceptor and elongate it. So from those results, when you ask what HG does Gout1 synthesize, we know it has the ability in vitro to synthesize the long polymer. We know it can make smaller sections, but we don't know yet from in vivo studies which of those polymers it synthesizes, because when we do studies of the mutant, and Melniot Mojo's heading those up, and we haven't finished those yet, so they're not published yet, what we've shown is it does not synthesize the bulk of Hg. It synthesizes some Hg tightly held into those walls. We think it's very interesting, probably evolved very early, and we're working on that. But we're still interested in what enzyme synthesizes the bulk of Hg. So the question is, if Gout1 doesn't synthesize it, which enzyme does? And so that leads to the question of what the function of these other gouts are. So we go back to this gout gene family. And I told you already that Gout1 and Gout7 are known Hg-GalATs. A series of people in the lab, and particularly Kristen, has now identified and shown proven enzyme activity that 5 other gouts have Hg-GalAT activity. I'm going to tell you now a story about 2 of those. When we're going to move back now to the goal of identifying genes that will reduce recalcitrance. So when we started looking at whether any pectins could reduce recalcitrance, we identified that there were 4 gouts that had impacts on biomass recalcitrance. And I'm going to tell you the story of Gout4 and at the very end a little bit about Gout9. So which gouts synthesize the different Hg domains? I'll give you a take-home first, and then we'll look at the biomass reduced recalcitrance story. So Gout1 and Gout4 both have the ability to synthesize polymeric Hg. Gout11 appears to be the gout that synthesizes Hg connected to Rg1. And Gout4 synthesizes the bulk of Hg in the cell. I'm going to tell you that story, as well as is required for the synthesis of the bulk of Rg2. Gout12 I won't talk about, but our data suggest us it synthesizes homogalacturinin required for xylan synthesis. So with that background now, you're prepared to understand what we have discovered in regards to a role of some of the gouts in biomass recalcitrance. So when we started in the Bioenergy Center, the question was, could we identify reduced recalcitrance genes? And the BEST, the Bioenergy Center, was a multi-institutional center of roughly 300 people scattered across the country, with the goal of reducing and understanding biomass recalcitrance. And there were 2 broad strategies. One was to improve microbes, which I'm not talking about, but what my personal research in BEST concentrated on was to identify genes whose modification would result in reduced recalcitrance. And there was a whole focus area dedicated to that, and a number of researchers have done some very nice work. And I'm going to tell you just 1.5 stories from that of some success we had. In BESC as a whole, if you ask which genes might you be able to modify to reduce biomass recalcitrance, you could, of course, imagine you might want genes involved in regulating cell wall synthesis, biosynthetic genes, genes that you could identify from natural variants that didn't affect cell wall biosynthesis. But we looked, based on transcriptomic analyses, if there were any pectin biosynthetic genes that might be interesting reduced recalcitrant candidates. So the reason pectin was an unlikely candidate is, as I mentioned before, pectin is relatively low abundance in poplar and switchgrass. So the way that you can analyze that is you isolate cell walls from wood or from aerial switchgrass tissue. You analyze the non-cellulosic carbohydrates, and what you'll see is, as one would expect, knowing a bit about the walls in those plants, in populus, the most abundant sugar is xylose, as in switchgrass, because of the glucuronoxylan or arabinoxylan hemicelluloses. And you say, well, how much pectin is there? Well, GALA is the diagnostic sugar, and that's GALA, and so you can see GALA makes up only about 6% of the sugar in poplar wood and about 1% in switchgrass biomass. And yet, when we used our knowledge of pectin synthesis and analyzed the transcriptomic data, we found some interesting gout candidates that we thought could be involved in formation of the biomass. And I want to tell you the story about that. This project was headed up by Ajay Biswal, and the first thing he did is, knowing a lot about wood development, wood starts with cells producing primary wall, they elongate, then there's a transition in cell wall synthesis to secondary wall synthesis, and eventually then you make your secondary walls, the woods. So if you analyze the transcriptome here, it was quite interesting that Gout4 and Gout12 were highly expressed there, and therefore we concentrated on them. And it was also surprising in switchgrass, aerial stems that Gout4 was highly expressed. So we said, well, let's study what happens if we modify the expression of Gout4. And that led to a very nice paper and the identification of a means to dramatically increase biomass yield and reduce recalcitrance in these two species. So what IG did is looked at the homologs of Gout4 in these species and in rice that were most closely related, the Gout4 homologs that were most highly expressed in those species, and working with other scientists in BESC, we made RNAi constructs and knocked down expression of Gout4 by 60 to 75% in populous switchgrass and rice. And what you see here, in populous switchgrass and rice, the black and the gray are vector controls and wild type, and the colors are all our individual transgenic lines, that this indeed confirms that the transcript was reduced by that level. So the first thing you do is you grow your plants in your greenhouses and do an analysis of the growth while you're trying to collect the biomass to do the recalcitrance analyses. And the first unexpected result was that in each species, the wild types on the left, an example of the transgenics on the right, in each species there was a dramatic increase in growth, an almost doubling of growth in the greenhouse of biomass yield in the greenhouse for switchgrass, up to 70% increase in rice and about a 50% increase in poplar. So now think about this a minute. You've got a very minor component in the walls in both these tissues. You're reducing transcript expression by 60%, and you're getting this dramatic increase in biomass yield. That's interesting. We then also grew the switchgrass lines in the field with a collaborative effort with Neil Stewart through BESC, and asked, would this phenotype be stable in the field after 3 years of field growth? Sorry, this pointer's a little bit slow. And what we found is, yes, year 1 was the establishment year, but in years 2 and 3, the black is a wild type, these are 3 individual knockdown lines, is that there was a dramatic increase in growth in the transgenics. It was stable in the field, and there was no negative impact of growing in the field, no negative impact of pathogens. So we had a 6-fold increase in biomass yield in the field. Was there any effect on recalcitrance, on this ability of the walls to be deconstructed? And it was modest, but it was real. So if you look at the ability to release sugar, you take the tissue, you dry it, you grind it up, and you incubate it with enzymes, and compared to the control, there was up to about a 13% increase in sugar release in the transgenic lines. If you combine that increase in sugar release with the increase in biomass yield in the field of the switchgrass grown in the field, there was a 7-fold increase in sugar release per plant. So these we felt were very important results, and provided a mechanism for increasing both plant growth and reducing recalcitrance. So then we said, well, what's causing this increase in biomass yield? And we did some analyses looking at the cell sizes. Leaf size was increased, leaf area was increased, phloem size was increased, and xylem size was increased. So there was clearly an increase in cell size that may have led to more total photosynthesis yield. So then we were very interested, how is this occurring? What is the mechanism for this improved growth and reduced recalcitrance in these switchgrass and poplar lines? So we did, and first, so you've got to step back. We've got a putative pectin biosynthetic gene. At this point, we hadn't tested Gout4 for enzyme activity. So the first thing you do is you look at the walls. Well, is there any change in the composition of those walls? And the first thing you do then is you take your biomass, grind it up, and make total cell walls. And when you do that and do a sugar analysis, the only sugar that was consistently released in all of these plants, switchgrass, rice, and poplar cell walls, was galactronic acid. It was reduced by 60% in poplar, 90% in switchgrass, and close to 80% in rice. So that's a dramatic reduction. You can only get so much information from total walls. In this case, it was very informative. So what you do when you want to begin to define what kind of polymers are affected, you take those total walls and you extract them with increasingly harsh solvents, ammonium oxalate, sodium carbonate, then 1 molar, K molar, 4 molar KOH, etc., to release different classes of polymers. And when you do that, you can do a sugar composition of each of those. And when you do that, again, the only sugar that was released in all of them was galactronic acid. It was reduced up to 97% in the extracted fractions, and even in that final fraction that you can no longer extract anything from, the Gallet was released. Showing that knocking down Gout-4 reduced the bulk of Gallet in the wall. We had the similar results when we did the analysis with poplar wood. That former one was switchgrass. Then you say, well, what sugar is it? It could be Gallet in RG1. It could be Gallet in RG2. It could be Gallet in HG. And you do a linkage analysis, and you see that it's reduced, the Gallet is reduced in the 4 linked position, telling you that it's homogalacturnan. So then this would suggest that perhaps Gout-4 is a galactorosyl transferase, but we had to prove that. So Gout-4, protein constructs of Gout-4, the same gene that was knocked down in each of those plants, were made and expressed heterologously in Nicotiana benthonianum cells. And indeed, that confirmed that each of those proteins from switchgrass, rice, and poplar, when expressed, had HG Gallet biosynthetic activity. The knockdown lines had less activity, and the product was sensitive to enzymatic degradation by endopolygalacturonase. So that showed the enzyme was an active, enzymatically active galactorosyl transferase. So how can we have these phenotypes? How can you have this dramatic increase in growth? How can you have the reduced recalcitrance with such a minor cell wall component? And now we're going to go back to the cross-linking. Remember, I told you pectins can be cross-linked in 2 different ways. So we asked, is the homogalacturnan salt bridge cross-linking decreased? Or is the borate diester cross-linking decreased? So you can measure that in various ways. You can measure the amount of calcium, and the left is always the wild type. Yes, the levels of calcium in the wall were decreased. There are antibodies that recognize this particular structure. Those were reduced in the transgenic. We measured the amount of boron in the walls. That was reduced. And we measured the amount of RG2 content based on diagnostic sugars, and those were also reduced. And so from that, we have a model for how we can get these dramatic effects on these biofeedstocks. Gout-1 synthesizes the bulk of HG and RG2 in the wall. When you not reduce its expression, you reduce the amount of those polymers. RG2 and HG are involved in these cross-links, and those cross-links were also reduced in the RNAi knockdowns. And so our model is the following. On the top would be the representation of the wall and the wild type. When you reduce the homogalacturnan and the cross-linkages, you get turgor pressure, you have less resistance to cell expansion. That can account for the increased cell growth associated with increased yield. In the same way, when you measure recalcitrance, you're looking at accessibility or extractability of the wall. With the reduction of these cross-links, the wall is more easily extracted, and you have better, easier access by the enzymes that degrade the wall. And so for these 3 mechanisms, these are the mechanisms we conclude are playing a role here in this reduced recalcitrance. All right, so I want to end with one more point. So clearly a knowledge of plant cell walls help us to attack a question and identify at least one gene that whose modified expression reduced recalcitrance. But now in the Center for Bioenergy Innovation, the question is, can one use natural variation to more rapidly redesign biomass for improved properties for biofuel production? And we had a very nice collaboration with Steve DeFazio, who is speaking later today, so I hope I don't misspeak too terribly about the results of this study. So Steve just published a very nice study that we collaborated with him on, in which they used a multi-trait genome-wide association analysis of a very large population of poplar that you already heard described earlier today, to try to identify, based on SNPs in those resequenced genome, variations that could be important for, in this case, leaf growth, among other characteristics. And the way they did that was they did both single and multi-trait genome-wide association of these greater than 800 sequenced individuals. And they measured 14 different physiological and morphological traits, one of them being leaf size. And from that analysis, which is a proof of concept that you can quickly do this, associate these SNPs with these traits to identify genes of interest to improve biomass feedstock, they identified 4 models with the single trait GWAS and 20 gene models with the multi-trait. And one gene that they found in each of those was Gout9. And then this shows you some of the data from that. And so the question was, it's nice to find a candidate that correlates with a phenotype, but is there additional evidence that it actually has a role in that phenotype? And so I want to remind you, here again is the gout gene family. I told you there were 4 genes we identified as associated with reduced recalcitrance. I told you about Gout4. We published on Gout12 a couple papers, and we're in the midst of trying to write up Gout9. So we had a whole set of data, including morphological measurements, and we had actually already measured the effect of knocking down Gout9 expression on leaf size. And so what you can see here, that when you reduce, in this case, by 50% transcript levels of Gout9, 40 to 50%, compared to the wild type, you had a dramatic increase in leaf size, length, and area. And so what this did is 2 different approaches, one based on expert knowledge and targeting genes based on transcriptomics, the other one's using these multi and singular GWAS analyses. The same gene was identified, and these data validate the functional role of Gout9 that's inferred by these single trait and multi-trait GWAS results. And so here I list the conclusions, and some of them are more specific, but the main one I'd like to make, I think, is that I think there's a power in combining fundamental understanding with these very powerful techniques of omics and GWAS methods. And finally, I think it's important to realize that the view of the wall that you see in models is simple, much more simple than the reality. And so if you're looking at genes that are identified in some of these omics strategies, I think it's very beneficial to interact with those people studying them to get the newest information about the current models. With that, I thank you for your attention. I'd be happy to address any questions. There was, oh, yeah.
Why are the plants bigger? Why are the plants bigger? Is it on? Did you change, is the division and elongation changed?
Cell size is in, it's larger, I think, cell size. There was also more, we didn't do a totally extensive analysis. There also seemed to be more total vascular number of cells, so at some point there was increased cell division there. So I think it's a combination of increased size, largely, with a bit more vascular tissue. But that level of study we need to do deeper. We were concentrating on the effects on the wall.
So somehow related to that, could you comment on whether there were major changes in morphology of leaves? For example, was it just bigger leaves, or actually the shape was somehow different? And also, could you comment on the root development, whether roots were bigger or more lateral roots or things like those?
Okay, so excellent questions. There was no obvious change in leaf morphology, but what we didn't do was to go and do minute measurements of all the parameters. You can kind of tell we're more biochemists than molecular geneticists. We didn't look at the roots. We should do that. We have a number of collaborations with a number of people, in addition to our lab, who are looking at various gouts on effects on growth. And so if you look at the gouts, most of them are expressed in most cell types, at least the ones we've looked at. So it's hard to say this gout has a role in this plant, this cell type at this time. But having said that, we need to do a lot more analysis of transcript analysis to look for specific roles in specific tissues at specific times. Your questions are excellent. As you've seen here, and I didn't tell you, you can modify several different gouts and get an increased growth effect, but we know that they appear to be affecting different polymers. I think what we will find, and this is just a hypothesis at my point, for some of these, my current view of what controls growth in the wall, and this is a very controversial area in the cell wall field. There was a hypothesis for a long time that you have your cellulose fibers, you have hemicellulose hydrogen bonding, and when you interrupt that, then you can get expansion. That's gone out of disfavor a little bit, and people are moving now, people like Cosgrove and other people are moving now to saying pectins seem to be involved, but there's not a good explanation, really. My hypothesis is that pectins are involved in multiple types of polymers in the wall, each one cross-linked, and that as you can affect that cross-linking, for example, you could imagine shooting out acid at one point and overcoming some of the calcium binding. You could have enzymes that remove the methyl esters and affect it. So I actually think this growth, what we may find in the end, is at least in large part a response of the cell walls inhibiting or enabling growth in response to turgor pressure. Some of it you may find specific effects in roots or some specific tissues, and it all needs to be studied at a greater detail. Right now we're trying to figure out the polymers.
I have a question here. Thank you for the talk.
I'm sorry, I don't know where you are. Oh, okay, yeah.
Hope the phone is on. Thank you for the talk, but I'm still trying to make sense of some of the results you presented, because it seemed to be a paradox to me. If the plants grow larger, it would require stronger cell wall and stronger vesicular structures. So if pectin is very important as a cross-linker, so that means it would require more pectin rather than less pectin. So how can you explain that?
Excellent question. I didn't make that clear. I was being pectinocentric. Of course, your cellulose fibers give incredible strength. Of course, your lignin in the wood is adding to that strength and affecting water conductivity. So the pectins are one component that may actually play a particularly important role in primary wall formation, but also at that point in time when those cells are dividing. I didn't tell you, there's no change in effect on cellulose and no effect on lignin. So those components would still be there, for example, in the wood to give that strength. I think the effect of pectin, okay, yes. You're going to have the bulk strength in the cellulose and in the, and lignin, and then the hemicelluloses. When we take those materials and grind them up, like we do when we do the sugar release assay, or that you might do when you're in a mill, you're already breaking some of that strength. So then you can imagine that other interactions, perhaps interactions that are involved in individual cell-cell adhesion as opposed to tissue strength, would take over. So I don't know if that answered your question, but that would be the best answer I could give you at present, and those are the results.